Students, now I am going to discuss another botany topic. It is the photosynthesis. In the morning session, I discussed one botany topic. It is the morphology in flowering plants. And in the second session, I discussed two zoology topics. One is the respiration and another is the excretion. In brief, okay, if you come to the college, when you come to the college, we will discuss each and every line in detail, okay. Now, another botany topic, it is photosynthesis. Before to explain this photosynthesis, let me discuss some terms concerned with this. The terms are, for example, metabolism. In the morning session, I told there are varieties of biochemical functions are going on. That biochemical functions can also be called as the biological functions. There are so many biological functions are there. Out of that, some biological functions are constructive processes, while some other biological functions are destructive processes. And both that constructive processes and destructive processes can be summarized as metabolic processes. There's a term, metabolism. Actually, this metabolism includes both catabolism and anabolism. Any destructive processes occur inside the living body can be called as catabolic processes, or you can say just catabolism. For example, respiration. Respiration is a catabolic process. Why it is called so? In that process actually, this in case of respiration, this glucose molecule, for example C6H12O6, this is a glucose. In respiration, this glucose molecule will be broken down into carbon dioxide and water. Of course, in that process, energy is liberated in the form of ATP. Right? Anyway, in that respiration process, this compound, for example, glucose, will be broken down. So, any such destructive processes can be called as catabolic processes or just catabolism. Respiration is an example for catabolism. Like that, there are so many such such functions are there. Anabolism, any destructive process, any constructive process can be called as anabolism. For example, photosynthesis is an example for anabolism. In that process, in that photosynthesis process, this glucose is constructed by utilizing the carbon dioxide and water, of course, with energy, ATP. So, photosynthesis is an example for anabolic process. Any constructive processes can be called as anabolic process. Right? Photosynthesis is an example for anabolic processes. Respiration is an example for catabolic process. So, in the living body, there are so many such catabolic and anabolic processes are going on. So, photosynthesis is one such anabolic process. Okay? So, both catabolism and anabolism constitute metabolism. All kinds of biochemical reactions, whether that biochemical reactions are constructive or destructive, all biochemical process in the living body can be called as metabolic processes or metabolism. So, that metabolism consisting of both the catabolism and anabolism, both the constructive processes and a destructive process. Right? So, this is the meaning of this metabolism, this catabolism and this anabolism. Okay? So, photosynthesis is an example for anabolic process. Right? It is one aspect. Photosynthesis in plants occurs in the chloroplast. Then what is that chloroplast? When you study the cell, when you study any eukaryotic cell, prokaryotic, I hope you know all these terms. For example, prokaryotic organism, prokaryotic cell. The cell with the false nucleus can be called as prokaryotic cell. False nucleus. For example, this is an organism, consider that. In this organism, this is a genetic material, maybe DNA, for example. Okay, the region of the protoplasm where this genetic material is present can be called as nucleus, right? So, nucleus is a region in the protoplasm of the cell where genetic material is present, nucleus. If this nucleus is surrounded by a specific nuclear membrane, then this nucleus can be called as true nucleus. True nucleus. So, nucleus which is surrounded by a specific nuclear membrane can be called as a true nucleus, right? The cell, the organism with the true nucleus can be called as eukaryote. This cell is a eukaryotic cell. If this organism, this is a eukaryotic organism, right? So, eukaryotic organisms are those which are having the true nuclei. True nucleus is one in which that genetic material is surrounded by a specific nuclear membrane. Eukaryotes. Then what are prokaryotes? Prokaryotic cell. If this is the cell, if this is the genetic material, so this area is called as a nucleus. Here, that nucleus is not surrounded by a specific nuclear membrane. If the nucleus is not surrounded by a specific nuclear membrane, then that nucleus is called a false nucleus. False nucleus. The cell with the false nucleus can be called as a prokaryotic cell. Prokaryotic cell. That organism, prokaryote. Prokaryotic organism, right? So, this is the main difference between the prokaryotes and the eukaryotes. Okay, prokaryotes having false nuclei, eukaryotes having true nuclei. True nucleus is one in which that nucleus is surrounded by a specific nuclear membrane, right? So, these are the difference between the prokaryotes and eukaryotes. Okay, when you consider any eukaryotic cell where we can see the presence of different organelles, for example, mitochondria, endoplasmic reticulum, Golgi complex, ribosome, lysosome, like that. There are varieties of different organelles are there. And each and every organelle is having specific function. For example, mitochondria. Mitochondria is also called as powerhouse of the cell because in that mitochondria, energy is synthesized. I will tell you that later. Like that, chloroplast. Chloroplast is an organelle present in all the green plants. In all the green plants, we can see in the cell of that green plants can be seen the chloroplast. How that chloroplast is present? You see, for example, that chloroplast is a double membranous structure. So, these are the two membranes present in the chloroplast. Chloroplast is a green color pigment, green color plastid. Right. So, this chloroplast is a double membranous structure. This is the outer membrane. This is the inner membrane. Right. This is outer membrane. This is inner membrane. Right. And this inner portion can be called as the stroma. Right. In this stroma, there are so many plate-like structures, disc-like structures, which are arranged one above the other. For example, this is one disc-like structure. This disc-like structure is called thylakoid. Right. and that thylakoids are arranged one above the other, okay, in a pile like this. So this single group can be called as granum. Granum is a collection of thylakoids. Right? This one granum, this another granum, there may be some more. So there are so many grana are present in the stroma of the mitochondria, chloroplast. Right? Each granum is a collection of thylakoids. When you consider a single thylakoid, each thylakoid is just like a tawa, like this. Here, the thylakoids are arranged in this manner. For example, if this one thylakoid, this membrane is called as a Fret membrane, intergranal membrane, interstromal membrane. You see, this is one thylakoid. This is a Fret membrane. Actually, this Fret membrane can also be called as the intergranal membrane because it connects the neighboring granum. Okay. While this one having the Fret membrane like this, Fret membrane like this, like this. In all these cases, they are present. Okay. And this Fret membrane or intergranal membrane, interstromal membrane actually connects the neighboring grana. Okay, right. In addition to this chloroplast may have some ribosomes also. Okay, some genetic materials also. Anyway, concerned with photosynthesis, we are talking about this chloroplast, about this thylakoid, about this granum. Now, in this thylakoid, we can see the presence of different photosynthetic units. That photosynthetic units can be called as the quantasomes. Photosynthetic units are called as the quantasomes. That quantasomes consisting of the photosystems. Photosystems are of two types. One is the photosystem 1 and another one is the photosystem 2. In each photosystems, different types of chlorophyll molecules are there in addition to different electron carriers. Okay, right. Concerned with chlorophyll molecules, I told you that chlorophyll molecules present in PS1 and PS2 in addition to different electron carriers. Right. Concerned with that chloroplast, in the photosynthetic units, there are different types. For example, chlorophyll A, chlorophyll A, chlorophyll B, like that chlorophyll C may be there. Okay. Chlorophyll molecules, different types of chlorophyll molecules. Okay. Carotenoids, another kind of chlorophyll molecules. Carotenoids. Okay. Of course, with different colors. Okay. Xanthophylls. Okay. Xanthophylls, light harvesting complex, etc. There are different such molecules are there. All these different molecules can trap the sun energy. You see, out of such so many pigments, different colored pigments, for example, this orange color pigment, blue color pigment, green color pigment, like that different colored pigments are there. Out of that, this chlorophyll A is also called as the reaction center. Chlorophyll A is also called as the reaction center. It is called reaction center because the actual photosynthesis occurs in this chlorophyll molecule, particularly in the chlorophyll A. Chlorophyll A specifically can be called as reaction center, where actually the photosynthesis function occurs, while chlorophyll B, chlorophyll C, carotenoids, xanthophylls, LSC, all these together can be called as accessory centers. Accessory centers. That different colored pigments actually trap the sunlight, send the same into this reaction center. So, except chlorophyll A, remaining all these different colored pigments, chlorophyll B, chlorophyll C, carotenoids, xanthophylls, all these together called as the axillary centers. Here, all these different pigments, they just trap the solar energy, send the same into this reaction center where actually photosynthesis occurs. Okay, so this is a brief information about the different colored pigments which are involving in photosynthesis, right? So as I told you, actual photosynthesis occurs in the chlorophyll A only, right? Remember that. Now come to the process. What is photosynthesis then? How does it occur? You see, photosynthesis, that biological term, is a combination of two words. One is the photos. Okay, another one is the synthesis. Photos means light. Light means sunlight. Synthesis means preparation. Preparation. What is preparation here? You see, all the green plants, by utilizing the raw materials like carbon dioxide and water, and by trapping the sunlight with the help of the chlorophyll molecule, they synthesize the carbohydrate. It is the C6H12O6, right? And in that process, this oxygen is also liberated as a byproduct. Actually, to be correct, in that process, even the water is also liberated. But sometimes this is omitted while writing the equation, right? So, photosynthesis means it is one of the anabolic processes in which all the green plants synthesize the glucose molecule or carbohydrate by utilizing the raw materials like carbon dioxide and water, and of course by trapping the sunlight with the help of the chlorophyll molecule is called photosynthesis, right? In that process, this oxygen is liberated as a byproduct. This is photosynthesis. This is the meaning, this is the definition of the term photosynthesis, right? In all the green plants, it occurs. It is nothing but, it is an anabolic process in which all the green plants synthesize the glucose molecules by utilizing the raw materials like carbon dioxide and water, of course, by trapping the sunlight with help of their chlorophyll molecule. Photosynthesis. Actually, that photosynthesis is also called as the redox process. Photosynthesis is also called as a redox process. It is called redox because it is both a reduction and oxidation process. Photosynthesis is both a reduction and oxidation process. In that process, this carbon dioxide is reduced into carbohydrate. Remember that this carbon dioxide is reduced into carbohydrate, glucose, while this water is oxidized into oxygen. So here carbon dioxide is reduced into carbohydrate, water is oxidized into oxygen. Because of this reason, this photosynthesis can also be called as the redox process. I think it is clear why that photosynthesis is also called as the redox process, right? You see, that entire photosynthesis process occurs both in presence and absence of sunlight. So that entire process can be divisible in two main steps. One is the light reaction. It is the first part of the photosynthesis, light reaction. And another one is the dark reaction. So, photosynthesis process occurs both in presence and in the absence of sunlight. The first part of the photosynthesis occurs in presence of sunlight, can be called as the light reaction. Second part of the photosynthesis which occurs in the absence of sunlight, not in the night, even in the daytime also. In the absence of sunlight, the second part of the photosynthesis occurs. It is called a dark reaction. It is not a night reaction. Remember that. Because this second part of the photosynthesis occurs in the absence of sunlight, even in the daytime also. That's why it is called a dark reaction, not night reaction. Remember that. Okay. Let us study what is that light reaction. Okay. Light reaction. You see, light reaction is also called as this light reaction. This light reaction is also called as the photophosphorylation reaction. Light reaction is also called as the photophosphorylation reaction. Why it is called so? Photophosphorylation reaction. Then what is phosphorylation? What is photophosphorylation? You see, phosphorylation, addition of phosphate. You can add that phosphate to any compound, any element, any material. Addition of phosphate, generally we can say, addition of phosphate is called phosphorylation. But here, addition of phosphate occurs to ADP, adenosine diphosphate. Okay, ADP plus inorganic phosphate gives rise to ATP. This is called phosphorylation. Addition of phosphate to ADP to produce ATP is called phosphorylation. I think it is clear. Okay, addition of phosphate to ADP to produce ATP is called phosphorylation. ADP, adenosine diphosphate. ATP, adenosine triphosphate. Here there are two phosphates. One more phosphate is added. That's why it will become adenosine triphosphate. Right? If this phosphorylation occurs in presence of sunlight, if this phosphorylation occurs in presence of sunlight, it is called photophosphorylation. Right? Photophosphorylation. In respiration also, we can see the phosphorylation, where in respiration, phosphorylation occurs in presence of oxygen. So, where it is called oxidative phosphorylation. Phosphorylation occurs in presence of oxygen is called oxidative phosphorylation. And that oxidative phosphorylation occurs in respiration process, while photophosphorylation occurs in photosynthesis. This is the main difference between the photophosphorylation and oxidative phosphorylation. So, concerned with the photosynthesis, we are talking about the photophosphorylation. I told you this light reaction is also called as the photophosphorylation reaction. In that light reaction, there is phosphorylation, means there is synthesis of energy like ATP, NADPH2, etc. Okay? Right. This photophosphorylation, or you can say this light reaction having some events. What are the events of light reaction? Number one, there is the excitation of chlorophyll molecule. One of the events, first event, one of the events of light reaction, or you can say photophosphorylation reaction, excitation of the chlorophyll molecule. Synthesis of ATP, and this synthesis of ATP can also be called as the evolution of, or can just synthesis of ATP. Third one is the synthesis of NADPH2 or NADH2. It is NADPH2 or NADH2. Next one is the evolution of oxygen. And another event is the photolysis of water. These are the events occur in the light reaction. Excitation of the chlorophyll molecules, synthesis of ATP, synthesis of NADH2 or NADPH2, evolution of oxygen, or you can say liberation of oxygen, right? Photolysis of water. Let us study one by one. Okay, actually that light reaction, as I told you, also called as the photophosphorylation reaction, right? That light reaction, or you can say that photophosphorylation reaction can be further classified into two kinds. They are the number one, cyclic photophosphorylation, cyclic photophosphorylation, and the second one is the non-cyclic photophosphorylation. So light reaction, or you can say photophosphorylation reaction, can be classified further into two types. One is the cyclic photophosphorylation and another one is the non-cyclic photophosphorylation, right? Cyclic photophosphorylation occurs in PS only. Cyclic photophosphorylation occurs in PS only, while non-cyclic photophosphorylation occurs in both PS1 and PS2. Right? Then what is cyclic? What is non-cyclic? Let us study that cyclic photophosphorylation. Okay? Cyclic photophosphorylation. Cyclic photophosphorylation. Phosphorylation. It is called a cyclic photophosphorylation because the same electrons which are coming out of the chlorophyll after moving through a series of electron acceptors come back to their original location. So because of the cyclic movement of that electrons, there is phosphorylation, there is photophosphorylation. Because of that, it is called cyclic photophosphorylation. For example, I told you the cyclic photophosphorylation occurs in PS1. Okay, that PS1 consisting of chlorophyll A 700, chlorophyll A 700, and that cyclic, that PS1, I told you, the cyclic photophosphorylation occurs in PS1, and this PS1 consisting of chlorophyll A 700 nanometer, okay, and there is a FRS, ferredoxin reducing substance. In your textbook, instead of this ferredoxin reducing substance, it is given that electron acceptors. FRS, ferredoxin reducing substance. There is another electron acceptor, FD, ferredoxin. There are different types of cytochromes. For example, cytochrome A, cytochrome F, cytochrome B, like that. Okay, there is a plastocyanin, 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 PC. So these are the different electron acceptors, electron carriers present in PS1. In addition to this, chlorophyll A 700. Okay, let us write that. Okay, in this cyclic photophosphorylation, you can come across with two events out of that 5. I told you there are 5 events. The first event is the excitation of the chlorophyll molecule. Second event is the synthesis of ATP. That 2 events can be seen in cyclic photophosphorylation. While in non-cyclic photophosphorylation, you can see all that 5 events. Let us study first one, cyclic photophosphorylation. You see, there is a chlorophyll A 700. This chlorophyll A 700 is present in PS1, I told you. When there is a falling of the light, when there is a falling of the light, light is nothing but light energy. It is nothing but solar energy. On receiving this solar energy, the electrons present here in the chlorophyll molecule are excited. This is the excitation of the chlorophyll molecule. Chlorophyll molecules are excited. on receiving this solar energy, this sunlight. Okay, then after receiving this solar light, sunlight, that light will be converted into chemical energy. Solar energy will be converted into chemical energy, and that chemical energy is carried over by the electrons of this chlorophyll molecule. Okay, that's how there is excitation of the chlorophyll molecule. Okay, so excitation of the chlorophyll molecule occurs because of falling of the sunlight on that chlorophyll molecule. After excitation, they trap that sun energy, solar energy. Then that solar energy will be converted into chemical energy. Then that chemical energy is carried over by these electrons present in this chlorophyll molecule. Okay, here there are a series of electron acceptors. This is one of the electron acceptor. I told you here there is FRS, then there is FD. These electrons, I told you, carrying the energy, remember that, carrying the chemical energy. Yes, then from this FRS or FD, or you can say according to your textbook, from this electron acceptor, that electrons are moved into electron transport system or electron transport chain. That electron transport chain consisting of cytochromes. I wrote there, cytochrome A, cytochrome F, cytochrome B, like that, different types of cytochromes are there. They are totally called as the electron transport system, electron transport chain. While the electrons are passed from this electron acceptor to this electron transport chain, 1 ADP plus P gives rise to ATP. You see, synthesis of ATP is an event. Excitation of the chlorophyll molecule is an event. Okay, so ATP is synthesized in cyclic photophosphorylation. Then it is transported into plastocyanin. From the plastocyanin, the same electrons come back to their original location. So after a cyclic movement of these electrons, or due to the cyclic movement of these electrons, there is the synthesis of ATP. That is why it is called cyclic photophosphorylation. Here phosphorylation occurs because of sunlight, because of solar energy, sun energy. Here that sun energy is present in the form of chemical energy. Okay, anyway, ATP is synthesized because of sunlight, and it is called photophosphorylation. And that photophosphorylation occurs because of this cyclic movement of the electrons, right? So this is the cyclic photophosphorylation. Phosphorylation occurs due to the cyclic movements of the electrons, due to cyclic movements of the electrons in PS1. Okay, is called cyclic photophosphorylation. So in this cyclic photophosphorylation, you can see two events. One is the excitation of the chlorophyll molecule, and another one is the synthesis of ATP. Right? So cyclic photophosphorylation is over. Come to the non-cyclic photophosphorylation. And I told you that non-cyclic photophosphorylation occurs both in PS1 and PS2. Right? In PS2, there is a chlorophyll molecule, chlorophyll A680. Chlorophyll A680 is the chlorophyll molecule present in photosystem 2. When the light rays once again fall upon this, these chlorophyll molecules are excited. Because of that excitation, they receive this solar energy. That solar energy will be converted into chemical energy. That chemical energy is carried over by the electrons. Actually, here per reaction, 4 electrons are coming out. But that much of depth information is not given in your textbook. Anyway, electrons are carrying this chemical energy. So here there is a electron acceptors. There is a compound called as the plastoquinone. leaving that, okay. So electron acceptors. From the electron acceptors, there is the electron transport chain, okay, electron transport chain. While these electrons are transporting from this electron acceptor to this electron transport chain, one ATP synthesized. ADP gives rise to ATP, right? Excitation of the chlorophyll molecule is an event. Synthesis of ATP is another event. Here, now, these electrons, instead of going back to their origin location, they move into PC. From that PC into chlorophyll A 700 of the PS1. I told you non-cyclic photophosphorylation occurs both in PS1 and PS2. You see here, the electrons which are come out of this chlorophyll A 680 after transporting through electron acceptors, instead of coming back, that electrons move into PS1. This is plastocyanin, chlorophyll A 700 is a PS1, from where they are transported into another electron acceptors. Electron acceptors. In that electron acceptors, there is a coenzyme. It is the NAD. NAD. So these electrons ultimately reach this NAD. Okay, that NAD combines with hydrogen, it will become NADH2, or you can say NADPH2. If this is the NADP. NADH2 or NADPH2 is also an energy. It is produced during non-cyclic photophosphorylation. Right. In this case also, the sunrise or sunlight can pass through even into this also. You see, I told you there are 5 events in the light reaction totally. In non-cyclic, we can see all the 5 events. Excitation with chlorophyll molecule, synthesis of ATP, synthesis of NADH2, 3 events are over. Remaining 2 events also occur during the non-cyclic photophosphorylation. It is called non-cyclic because the same electrons which come out of this chlorophyll A680 do not come back. You see, these electrons, instead of coming back to this molecule, these electrons, after moving through a series of electron acceptors, ultimately it reaches the NAD. It is not a cyclic, it is a non-cyclic. Because of this non-cyclic movement of the electrons, there is synthesis of NADH2, synthesis of ATP, nothing but phosphorylation. It is called non-cyclic photophosphorylation. Photophosphorylation occurs because of non-cyclic movements of the electrons. Non-cyclic movement of the electrons both in PS1 and PS2. I hope you have understood. This is non-cyclic photophosphorylation. It is called so because the same electrons which come out of the chlorophyll A680 do not come back. Do not come back. That's why it is non-cyclic. Okay, if they are coming back, they become cyclic. They do not come. Instead of coming back, they go to the NAD of PS1. It is included, it is present now in the NAD. That will become NADH2. It's an energy. NADH2 is also energy, just like ATP. So this is the non-cyclic photophosphorylation. Right? And I told you in the beginning, in the non-cyclic photophosphorylation, all the 5 events occur. But here, I have shown only 3. Excitation of the chlorophyll molecule, then synthesis of ATP, synthesis of NADH2. What about photolysis of water? What about liberation of oxygen? You see, during non-cyclic photophosphorylation, that photophosphorylation and the liberation of oxygen also occur. Why? You see, actually, per reaction, 4 electrons coming out. If the electrons are coming out continuously, because of the loss of the electrons, there may be there will be the collapsing of the chlorophyll molecule in the plant body. But it doesn't occur. There will be no question of collapsing of the chlorophyll molecule because that electrons are supplied from outside into this molecule to make up the loss. That electrons, they are lost, but there is a continuous loss of the electrons. That loss is made up by supplying that electrons from outside. From outside means from where? During photolysis of water. Right? Then what is a photolysis of water? Photolysis of water. Then what is photolysis? Photo, sunlight. Lysis, breakdown. The process of breaking down of water molecule into ions like hydrogen ions and hydroxyl ions. This water molecule will be dissociated into hydrogen ions and hydroxyl ions. Of course, in that process, electrons are also liberated. Of course, in that process, oxygen will also be liberated. I will tell you that later. Electrons will be liberated during this photolysis of water. So, photolysis, the process of breaking down of water molecule into ions like hydrogen ions and hydroxyl ions. Right? So, this photolysis of water. In that photolysis of water, these electrons also liberated. These electrons are transported to the PS1, chlorophyll A680, PS2 of chlorophyll A680. That's how the loss is made. Okay? So, this photolysis of water. 4 events. Liberation of oxygen. You see, in further process, there are 4 hydroxyl molecules per reaction actually. 4 water molecules are dissociated. 4 hydrogen ions, 4 hydroxyl ions are produced. Out of that 4 hydroxyl ions, there is a protein called as the Z protein. To that Z protein, these 4 hydroxyl molecules are attached like this. Ultimately, it's called Z protein complex. It is a depth information. It is not necessary, I think. You see, all these 4 hydroxyl molecules are attached to this Z protein complex. Now, O plus O will become O2. Remaining O and H, water is liberated. 2H2O and oxygen. So, from these hydroxyl molecules, this oxygen is liberated. So, during photosynthesis, O2 is liberated and that O2 is coming from water. This O will become O2, not from carbon dioxide. CO2 plus water gives rise to C6H12O6 like that. So, O2 liberated during photosynthesis is not coming from carbon dioxide, but it is from water, hydroxyl molecules. Right? So, during photosynthesis, O2 is liberated and that O2 is coming from water. Coming from water, not from CO2. Remember that for CET question. So, here you see this is a photolysis of water. So, 4th event, and because of the photolysis of water, this oxygen is also liberated. So, that in non-cyclic photophosphorylation, you can see all the 5 events. Excitation of the chlorophyll molecules, synthesis of ATP, synthesis of NADPH2 and liberation of oxygen and photolysis of water. This is the non-cyclic photophosphorylation. Here, this water molecule is ionized into ions. That's why this photolysis of water can also be called as the photoionization of water. Photolysis of water is also called as the photoionization of water. In that process, that molecule will be ionized into ions like hydrogen ions and hydroxyl ions. Ultimately, from that hydroxyl ions, O2 is liberated. Water is also liberated. So that during photosynthesis, water is also liberated. This actual equation of photosynthesis is this: CO2 plus water gives rise to C6H12O6 plus O2 plus water. This is the actual equation of photosynthesis. But sometimes, in most of the times, we neglect this. We write only these two. Okay? So here, this O2 is liberated as a byproduct. Oxygen is the byproduct of photosynthesis. Remember that. Okay? Now you can balance this equation that you know, I think. Okay? So this is the photosynthesis. Okay? So up to here, we studied about the light reaction only. Light reaction only. Only the first part of the photosynthesis. In that, what are the products of light reaction? You see, these are the products of light reaction. Number one, synthesis of ATP. Synthesis of ATP, one of the products of light reaction. Second one, synthesis of NADPH2. You see, this NAD is nothing but the full form of that NAD, nicotine amide. N means nicotine amide. Up to that, nicotine amide. A, adenine. D, dinucleotide. So NAD means nicotinamide adenine dinucleotide. P, phosphate. H, hydrogen. It is like that. Okay? So light reaction is over. In that light reaction, what are the products of light reaction? Number one, synthesis of ATP. Number two, synthesis of NADH2 or NADPH2. Number three, liberation or evolution of oxygen. These are the products of light reaction. And this oxygen will be liberated directly into the atmosphere while it is producing. As soon as it is producing, it will be released to the atmosphere. Only these two energy are retained in the cell, in the chloroplast. Okay, so in the entire light reaction, only half of the processes is completed. In the entire light reaction, these are the products. Out of these three, this is liberated into the atmosphere. These two are retained. And these two are utilized for dark reaction. Light reaction is over. You see, light reaction is over, but CO2 is not utilized according to equation. CO2 plus water gives rise to C6H12O6, like that. Light reaction is over, CO2 is not utilized. So, in the entire light reaction, what happens? These two energies are produced. These two energies are also called as the assimilatory power. Assimilatory power, which are utilized for dark reaction. Both ATP and NADH2 are called assimilatory powers, energies. They are utilized for dark reaction. This is already liberated into atmosphere. Anyway, if the question is asked like what are the products of light reaction, you have to write all these three: synthesis of ATP, synthesis of NADPH2 and the evolution of oxygen. As I told, it is liberated into atmosphere. These two are retained and these two are the energy, and that two energy are called as the assimilatory powers, and that two energy are utilized for dark reaction. Remember that. Okay? So light reaction is completed. I hope all of you have understood very clearly. Okay? Come to the dark reaction. Dark reaction. Second part of the photosynthesis. This dark reaction can also be called as the synthetic phase. Light reaction, preparatory phase. The necessary energies are prepared. Preparatory phase, light reaction. Dark reaction, it occurs in absence of sunlight, and that dark reaction also called as the synthetic phase. It is called so because in that dark reaction, that glucose is synthesized. Okay? Two names, dark reaction or synthetic phase. This dark reaction is also called as the Calvin cycle. Calvin cycle, because Melvin Calvin, scientist who studied this very well in detail. So this dark reaction also called as the Calvin cycle. Right? Calvin cycle is also called as the C3 pathway cycle. It is called so because in that reaction, 3 carbon compound, it's actually PGA, phosphoglyceric acid, is produced. The first stable compound produced during dark reaction is this 3 carbon compound. The name is phosphoglyceric acid, or you can say phosphoglycerate, you can say, right? So because of that, it is also called as the C3 pathway cycle. 3 carbon compound is the first stable compound produced in that dark reaction. So that name is given. It is well studied by Melvin Calvin. That name is given, Calvin cycle. In that process, the glucose is synthesized. So it's a synthetic phase. It occurs in the absence of sunlight. So it is called a dark reaction. Right? And that dark reaction consisting of these 3 steps. One is the carboxylation. Okay. Carboxylation. Then glucose formation. Carboxylation and glucose formation. 3, regeneration. It has one more name, I will tell you that later. Regeneration. Okay, these are the 3 steps present in the dark reaction. Carboxylation, glucose formation, then regeneration. Right? What is this carboxylation? It is just like phosphorylation. Phosphorylation means addition of phosphate to ADP to produce ATP. Carboxylation, addition of carbon dioxide. Addition of carbon dioxide to a compound, it is called as the RUDP. It's a 5 carbon compound. 5 carbon compound. Okay? So ribulose diphosphate. Ribulose diphosphate is a 5 carbon compound present in the chloroplast, right? And that 5 carbon compound combines with this single carbon compound, it will become a 6 carbon compound. That 6 carbon compound is unstable, so it is immediately dissociated into 2 compounds with 3 carbons each. They are the PGA. This is the carboxylation. Addition of carbon dioxide to RUDP to become PGA is called carboxylation. Second step can also be called as a reduction. I told you that reduction is nothing but synthesis of glucose. Okay? I hope you have understood the meaning of this carboxylation. Addition of carbon, not carbon, remember that. Addition of carbon dioxide to RUDP to become PGA. Right? Carboxylation. RUDP, ribulose diphosphate. It is a ribulose diphosphate. Okay? It's a 5 carbon compound. That 5 carbon compound combines with this single carbon compound, it will become 6 carbon compound. That 6 carbon compound is very unstable. So immediately it is dissociated into 2 compounds with 3 carbons each. It is the phosphoglyceric acid, or can say phosphoglycerate. Right. Then reduction, stage. I told you this is called glucose formation. Reduction. In this second step, that PGA will be reduced into trioses, that trioses, 3 carbon compounds, will become glucose, fructose, like that 6 carbon compound. And this reduction of PGA into this 6 carbon compound occurs in presence of that ATP and NADH2, which are produced during light reaction. I told you these two are called as the assimilatory powers, which are produced during light reaction. Now that two are utilized for reduction. That two energy, both the energies are utilized for reduction of this PGA into this glucose. This is reduction. Here PGA is reduced into glucose. Ultimately, it's a glucose formation process. Third step is the regeneration. What is that regeneration? It is nothing but reproductions of this RUDP, which is utilized in the beginning of the reaction. Okay, so from here to here it is the carboxylation, from here to here this reduction, and from here to here this is the regeneration. And that regeneration also occurs because of the utilization of that ATP, that will become ADP. This is the dark reaction. This is the Calvin cycle. Okay, from here to here this is carboxylation. We can write here, for example, carboxylation, for example. This one, reduction. Okay, reduction. This one, regeneration. Carboxylation, reduction, regeneration. So regeneration is nothing but it is a re-obtaining of the compound RUDP, which is utilized in the beginning of the dark reaction. Okay, it occurs by utilizing this glucose only. There are so many reactions are there, it is not given in our syllabus. Okay, simply it is given like this only. So I told. So this is the Calvin cycle. This is the dark reaction. This is the Calvin. Full name is Melvin-Calvin cycle. Okay, so this is about the photosynthesis. Ultimately this glucose is produced. Photosynthesis is over. That photosynthesis, I told you, this particular reaction is also called as the C3 pathway cycle, because in this path the 3 carbon compound, it is PGA, is the first stable compound obtained in this reaction. It is called C3 pathway cycle separately because there are some plants, they produce the 4 carbon compound where 3 carbon compound is not produced, where it is called 4 carbon, is a 3C pathway cycle, where it is a 4C pathway cycle. 4 carbon compound is the first stable compound produced. For example, OAA, oxaloacetic acid, it's a 4 carbon compound. Oxaloacetic acid is a 4 carbon compound produced in some xerophytic plants. Xerophytic plants, they are the plants which are growing in the dry area with less content of water. Okay, like cactus for example. Okay, Apansia. Like that, there are so many plants are there where C4 pathway cycle occurs. C4 pathway cycle. And that 4 carbon compound is the oxaloacetic acid. It's the first stable compound produced in C4 pathway cycle in some cases only. But in majority of the green plants, C3 pathway cycle occurs. Right. One more important information you have to know that C3 pathway cycle occurs because of presence of an enzyme. It is the Rubisco. Rubisco is an enzyme. It is the only enzyme present, protein enzyme present in the entire biosphere. Biosphere, it is the area of the earth where the living organisms are present, both plants and animals. In the entire biosphere, the largest amount of protein enzyme present is the Rubisco. This Rubisco enzyme is the only enzyme which is involved in C3 pathway cycle. C3 pathway cycle. Right. What is this Rubisco? Actually, it's an abbreviation, not the full form. Rubisco, the full form of this enzyme is the ribulose. Ribulose. Are you? Biphosphate. Sorry. Biphosphate. Carboxylase. Carboxylase. Oxygenase. This is the full form of that enzyme, Rubisco enzyme. Ribulose, biphosphate, carboxylase and oxygenase. This enzyme is called as the carboxylase and oxygenase because sometimes it accepts carbon dioxide or carbon, you can say, sometimes oxygen. I will tell you that later. Because of that, that name is given. So Rubisco is the enzyme which is involved in C3 pathway cycle. In C4 pathway cycle, another enzyme is involved. It is called as the phosphoenol pyruvate, PEP. Phosphoenol pyruvate. This is the enzyme. Phosphoenol pyruvate is an enzyme. PEP, phosphoenol pyruvate, is an enzyme which is involved in C4 pathway cycle. Rubisco is an enzyme involved in C3 pathway cycle. These are the enzymes, proteinaceous enzymes. Okay, so complete photosynthesis process is over. Okay, so photosynthesis occurs both in presence and in the absence of sunlight. It is called light reaction, dark reaction. After completing both of them, we can see the productions of the glucose, right? You see, any biological process occurs by the influence of some factors. That factors may be internal, may be external. For example, what are the important external factors involving in photosynthesis? If the question is asked so, these are some of the important external factors involved. For example, water, sunlight, carbon dioxide, temperature. These are the important external factors involving in photosynthesis. They are all influencing the rate of photosynthesis. These are the external factors. Like that, there are so many internal factors which are also involved influencing the rate of photosynthesis. For example, number of chloroplasts, one of the internal factors. Number of chloroplasts. Leaf size, okay. Like that, there are so many different types of enzymes, etc. So ask the question about what are enzymes. You see, enzymes are the biocatalysts. They are catalysts. In chemistry, you are studying what is a catalyst. Catalyst is a substance. It accelerates, either it increases or decreases the rate of reaction. Okay? Anyway, it accelerates. So, catalyst is a substance which accelerates the rate of the chemical reaction. But at the end of the reaction, it remains unchanged. It remains as it is. That substance is called a catalyst. In the living body, there are so many such chemical reactions. They can be called as the biochemical reactions. So the rate of the biochemical reactions are controlled over by, or the rate of that biochemical reactions is increased or decreased in presence of a substance that can be called as a biocatalyst. That biocatalyst is nothing but the enzymes. Enzymes are the biocatalysts. biocatalysts, they accelerate the rate of biochemical function. In the morning session, I told about the role of the enzyme. It is the carbonic anhydrase. I told carbonic anhydrase. Carbonic anhydrase is an enzyme which increases the rate of the biochemical reaction, rate of the reaction between the carbon dioxide and water. In the morning I told the reaction between these two is very slow. Only about 200 carbonic acid H2CO3 are produced. Carbonic acid molecules are produced. About only 200 such molecules are produced in 1 hour. It takes about 1 hour, 60 minutes to produce these 200 carbonic acid molecules in the absence of enzyme. But in presence of this enzyme, carbonic anhydrase, the rate of the reaction between these two will become very fast. How much it is fast? It is increasing. You see, per second 6 lakhs of carbonic acid molecules are produced. So you can imagine the role of the enzymes. So enzymes are the biocatalysts. Usually they increase the rate of reaction. You have studied so many such enzymes last year while you studying the digestion, for example. Digestive enzymes are there. Carboxylases, for example, salivary amylase, pancreatic amylase, pepsin, renin, like that there are so many enzymes are there. They accelerate the rate of the digestion process in that particular case. Okay. So these are the enzymes. So in photosynthesis also there are some enzymes which are involved. They are one of the internal factors. Remember that. Like that, if the leaf size is large, if the leaf lamina is large, automatically number of chloroplasts will be increased. If the number of chloroplasts will be increased, rate of the photosynthesis will also be increased. It is like that. So like that there are so many such internal factors are there. Okay. So these are the external factors: water, sunlight, carbon dioxide, temperature. Okay. Water is one of the important factors which can affect the rate of photosynthesis. You see, if the sufficient amount of water is present in the plant body, for example, all the cells in the plant body will be in the turgid condition. What is this turgid condition? You see, this is the turgid condition. If sufficient amount of water is present inside the protoplasm, this cell will be in the swollen condition. That swollen condition can be called as the turgid condition. Right. That turgid cell can perform any biochemical reactions at a good rate. So rate of photosynthesis is increased with increasing in the sufficient amount of water in the cells, because that water maintains the turgidity of the cell. That turgid cell can undertake the photosynthesis effectively. If the water content is decreased, there is a shrinking of the protoplasm of the cell. Shrinking. Okay, it is called as the plasmolized condition. Shrunken condition. If the protoplasm is shrunken, rate of biochemical reactions, any biochemical reaction, the rate of any biochemical reaction will be decreased. That's how water is playing an important role. Not only concerned with the turgidity, even for photosynthesis to produce the glucose, this water is necessary. Because of the presence of water, there is synthesis of carbohydrate. Okay? Of course, there is liberation of oxygen. That's how this water is playing an important role in deciding the rate of photosynthesis. Sunlight is also one of the important factors. If the sunlight, if there is a bright sunlight, concerned sunlight, minimum, optimum and maximum. At minimum sunlight, rate of photosynthesis function starts. Photosynthesis function starts at minimum. Photosynthesis is increased. Rate of photosynthesis is increased with increasing in the sunlight. So, if the sunlight is very bright, the rate of photosynthesis will also be increased. So, rate of photosynthesis will be increased with increasing in the intensity of the sunlight. Intensity of the sunlight. Intensity, quality, okay, duration, these factors of the sunlight playing an important role. For example, intensity, if the quantity of sunlight is more, rate of photosynthesis is also more. Intensity, brightness, okay, quantity. Quality, actually in the sunlight there are different rays are there. Out of that, both blue and red rays are very essential for good rate of photosynthesis. Both blue and red rays are very essential for good rate of photosynthesis. If the sunlight consisting of large amount of blue and red rays, it is a quality of the sunlight which increases the rate of photosynthesis. Like that, duration, you see, in the year, in the entire year when we consider, in some months, day is long. In some months, night is long. In some days, both night and day are in equal, 12 hours, 12 hours. So, there are some plants, day long plants, day short plants, day neutral plants. Day long plants, these are the plants, they need more than 12 hours of sunlight per day. For example, sugarcane, day long plants. So, for sugarcane, it needs more than 12 hours. So, duration of sunlight should be more than 12 hours. Like that, there are some plants, they are the day short plants. They just need 8 to 10 hours per day. There are some plants, they need equally 12 hours night, 12 hours day. Like that, the sunlight playing an important role in deciding the rate of photosynthesis and even other functions also. Like the temperature also, one of the important factors. Concerned with the temperature, the photosynthesis process begins at a minimum. Okay, minimum. Rate of photosynthesis is increased to optimum at optimum temperature. Rate of photosynthesis is maximum if the temperature is also maximum. So, photosynthesis begins at minimum, increases at optimum, increases very lot at maximum. So, minimum, optimum and maximum. These are three levels of temperature which are necessary. Beyond maximum, rate of photosynthesis is decreased. Even the sunlight also. If the sunlight is beyond maximum, there will be the burning of the chlorophyll molecules, entire leaf. Like that, consider the temperature also, at a minimum, it starts, it increases at optimum, rate of photosynthesis will be very maximum at the maximum temperature. Beyond maximum, below minimum is not good for photosynthesis or any biochemical functions. Rate of photosynthesis will be decreased below minimum and beyond maximum temperature. Like that, temperature is also playing a very important role. Actually, these enzymes are very active at optimum temperature. If the temperature is increased, these enzymes will die off. They will be died. Below minimum, they cannot start the function. Below minimum temperature, enzymes cannot become active. So, enzymes become active with increasing the temperature. They are at maximum temperature. If the temperature is crossing the maximum, the enzymes will be died off. Without enzyme, no biochemical function. That's how there are different, so many interlinkings with one another. That's how this water, the carbon dioxide, sunlight also, all these different things are playing very important role. Okay, like that carbon dioxide also. 0.03 at this concentration, rate of photosynthesis is good. Up to 0.5% it is okay. Beyond this percentage of carbon dioxide, rate of photosynthesis will be decreased. That's how even carbon dioxide also playing a very important role. Okay. Anyway, generally we can say that any biochemical function is controlled over by several factors. When any biochemical function is controlled over by several factors, the rate of that function will be decreased with the minimum factor. With the minimum factor. It is called a law of limiting factor. Law of limiting factor. This law of limiting factor was proposed by a scientist known as Blackman. Law of limiting factor was proposed by Blackman. According to law of limiting factor, when a biochemical process is controlled over by several factors like water, temperature, light, etc., enzyme, etc., the rate of that function will be decreased with the least factor. The rate of that function will be decreased with the least factor. With the least factor. For example, consider that there is a bright sunlight, there is a temperature, there is water, but there is a very less oxygen or very small amount of enzyme. Because of the presence of less concentration of carbon dioxide, even though there is a large amount of water, temperature, sunlight, etc., rate of that function will be decreased. Like that, there is a sufficient amount of carbon dioxide, temperature, water, etc., but no sunlight. Very least amount of sunlight is there, dim light. So rate of photosynthesis will be decreased. Rate of photosynthesis will be decreased because of very lesser amount of sunlight. This is the theme of this law of limiting factor. According to this, the rate of any biochemical function will be decreased with a least factor. The factor which is very less. With the least factor, the rate of biochemical function will be decreased. This is the law of limiting factor proposed by that Blackman. Okay, so this is the information about the different factors, both internal and external factors which affect the rate of photosynthesis, in general, rate of any biochemical function. Students, I hope this entire photosynthesis process is completely explained. I think you have understood. Okay? Okay, thank you.